In this talk, we will follow the progress of the earliest steps in embryology, from fertilization to formation of the bilaminar embryo. We're going to pick up right here at the very moment after fertilization has occurred. We can see that a spermatozoa has just entered the oocyte. It is punctured through the protective barrier of cells and the zona pellucida to reach the oocyte and its cytoplasm. As soon as a single spermatozoa has entered the cytoplasm of the egg, it sets off a chain reaction of events that prevent additional spermatozoa from making it in. At that point, the one male pronucleus consolidates inside the cytoplasm and then begins to associate with the female pronucleus. Once these two fuse and come together, we have now got the genetic material making up a brand new and distinct individual. At this point, we have the diploid complement of chromosomes that can be used as the blueprint for all the subsequent formations and changes that will bring about the human being that can grow from this single cell. As these nuclei have formed, a series of events can begin that have been on hold for quite a while in the egg. In particular, mitotic division can start taking place again, and the chromosomes can go through the whole process of metaphase, anaphase, telophase, and produce subsequent generations of cells that are genetically similar, in fact identical, to the progenitor cells. Roughly 30 hours after fertilization, the first split is going to occur and have two daughter cells take the place of the single zygote. Now at this point, subsequent divisions are going to occur, and the resulting cells will not grow appreciably. In fact, they'll take up as much space as the original single-celled zygote did. So they will get smaller and smaller even as they become more numerous. At this point, we're going to refer to the entire assemblage as a morula, a collection of cells. And we're going to take a look at the 9-cell stage, roughly 2.5 days into development. At this time, we've got something about the same size as the zygote, and it's going to consist of a group of cells clustered in the center, the inner cell mass, and a surrounding outer cell mass. During this time, the protective barrier that existed around the developing zygote and morula, the zona pellucida, begins to break down, and fluid begins to permeate into the spaces between these cells. Thereafter, we're going to go a bit further to a stage of development called the blastocyst. The fluid that is moved into the spaces between the cells of the morula creates a cavity called the blastocyst cavity, and the entire assembly of fluid and cells is referred to as the blastocyst. Now, the early blastocyst has roughly 58 cells, and it will continue to develop to roughly the 107-cell stage after 5 days, which is going to be called the late blastocyst. It looks about the same, with a fluid-filled cavity surrounded by cells on the outside and a cluster of inner cells pushed off to one pole of the blastocyst. The inner cell mass and outer cell mass will form very distinct structures. As we proceed from the morula to the blastocyst and further on into development, we'll see that the outer cell mass transitions into a set of structures called the trophoblast, and the inner cell mass is going to become the embryoblast, and the embryoblast is what will generate all the tissues of the actual embryo and fetus, whereas the trophoblast will become the supporting tissues of the placenta and the umbilical cord. Now, let's take a look at how development proceeds from the blastocyst into the bilaminar embryo, and during this time, we have the embryo moving into the uterine wall from days 7 to 15. Initially, the inner cell mass, which will form the embryoblast, subdivides into a thin layer of cells in contact with the blastocyst cavity called the hypoblast, and a taller group of cells further away from the blastocyst cavity called the epiblast. The epiblast and hypoblast cells have distinctive fates. The hypoblast cells are going to be proliferating to surround the blastocyst cavity. They are small and cuboidal, whereas the epiblast cells grow taller and taller and are going to be the cells that actually create the entirety of the embryo from this stage on. Now, moving into the uterus, the developing blastocyst is going to have these epiblast cells and hypoblast cells do a few different things. In particular, a small cavity is going to form in the epiblast cells called the amniotic cavity. Now this tiny little pinpoint of a cavity may not look like much right now, but it will be the cavity that surrounds the entirety of the embryo and fetus, forming the water that supports and nurtures it throughout development. So the amniotic cavity will eventually surround the entire fetus. As we go further and further into the uterus, the hypoblast cells are going to proliferate and spread out across the blastocyst cavity. Once they've completely done so and extended what's sometimes called Heiser's membrane throughout that space, that entire cavity will transition to become the primary yolk sac. Moving a little further along, the primary yolk sac and embryo become separated from the surrounding trophoblast cells by a distinctive set of cells called the extraembryonic mesoderm. This extraembryonic mesoderm will not contribute anything of substance to the actual embryo, but allows it to develop fully inside the uterus and separates it from the placenta. This happens because some cavities are going to start appearing inside the extraembryonic mesoderm, and these cysts or little cavities are going to form, fuse, and become what's known as the extraembryonic coelom, a space that forms outside of the developing embryo, and as it enlarges and completely wraps around the yolk sac and the embryo, it will form what's called the chorionic cavity. Initially, the chorionic cavity will surround the embryo and only much later be replaced by the expanding amniotic cavity. So as we move a little further along, the expansion of the chorionic cavity pinches the yolk sac in such a way that a portion of it moves away from the embryo towards the opposite pole of the developing embryoblast. And the part of the yolk sac that remains in contact with the embryo is going to be distinct, and it's going to form what's called the definitive yolk sac. The portion that moves away is going to form remnants of the primary yolk sac called exocoelomic cysts. Now the secondary or definitive yolk sac stays in contact with the epiblast cells that are developing into the embryo and have an ever-enlarging amniotic cavity within. Now here, as the chorionic cavity and the placenta has grown larger and larger, we can see that the extraembryonic mesoderm is lining the developing placenta and chorionic cavity and is only connected to the developing embryo by a single small connecting stalk. That connecting stalk is what connects the developing bilaminar embryo to the developing placenta, and that is what will become the umbilical cord. Thank you very much for your attention, and I'll see you during our next talk.